Hello, welcome to the ASAP Protocol Particular series. For today's episode, I have with me Garardo Ongari, who's a postdoc in the Blandini lab for Team Schapira. Hello.
Hello. As Devin said, I'm a postdoc researcher at the Mondino Institute in Pavia, and I'm a part of Team Schapira. And in particular, my laboratory is involved in the study of patients with glucocerebrosidase mutations. And we are involved in the analysis of samples from blood of the patients. We are studying the lysosomal pathways, and we are searching for biomarkers connected to the gut barrier function. So this is what my laboratory is working on.
Awesome, thank you. So for today's episode, we are going to focus on your protocol isolation of PBMCs from whole blood. And so you already told us a little bit about the project that you're working on, but maybe you can go into some more detail about how you're actually using this protocol for the project.
Yes, so we are searching for biomarkers, and we are working mainly on PBMCs or peripheral blood mononuclear cells and plasma samples. So it's important to have a standardized protocol to isolate these cells. And that's the reason why I've written this protocol. And so we are working with a different center, and it's important to have a standardized protocol to share among the centers. And we tried to write this protocol to optimize it to reach a good amount of cell isolated, and to minimize the sample contamination. So our protocol is based on the classical separation of PBMC by gradient density, so the stratification on Ficol. And this protocol is well, not so difficult, but it has some critical steps that may be difficult for beginners. So this is why I decided to write it for a standardized version of the protocol, and to make it easier to perform it.
Yeah, you're right. Definitely having these protocols will help with replicability across different labs and, reproducibility within your own group as well. Before we get into like the specifics on the steps, I was wondering about the sample. Like if for this protocol, if you can use fresh sample or if only frozen whole blood, and if you can use frozen, if there's anything like special that you have to consider when utilizing that.
Yes, no, this protocol is used on a fresh samples, because it's not a good idea to use it on frozen samples, because when you froze the samples and then you freeze them, the cells will lyse, and so this will make the certification almost impossible. So an important thing is that you have to process your samples as soon as possible, because if you delay the process, you will increase the risk of sample contamination from red blood cells. There is some way to solve this problem, like using a red blood lysis buffer, but every time you repeat this step, you'll you'll decrease your sample yield. So it's better to try to process your sample immediately than to wait and use then a lysis buffer.
That's good to know. So when people are doing this experiment, they really need to think about the timing of actually obtaining these samples and then doing the experiment right away. Um, so I see that the protocol is split into two major parts. One is isolating plasma and then isolating PBMCs. And so thinking about the plasma part first, I was wondering if you could detail some of the most critical steps that people should be aware of.
Yes, in reality, the plasma step, the isolation of plasma is the easiest step probably of the protocol because the only mistake that you can make during the isolation of plasma is to retrieve some of the cellular part. So after the centrifugation, you'll have two parts, the plasma part in the upper part of your Falcon tubes and the cellular part below. And it is easy to avoid this problem. You just have to stop before reaching the plasma part, the cellular part, and it's not a problem if you leave some plasma because the plasma, the remaining plasma won't will ruin your following isolation of PBMCs. So just leave it and don't risk to take some of the underlying part just to take all the plasma. Is not a problem if you leave the plasma part.
That's really good to know. And so if
There
we,
are also other things about the plasma. You have to think about which analysis you perform before you choose how to take the blood samples because the use of different protocols can require the use of different anticoagulant like heparin or DTA, and some protocols also require to isolate the plasma samples at low temperatures. So it's important to take care of the analysis that you're going to perform before, even before taking the blood samples.
okay, so it sounds like people should be very prepared and consider all the next steps before
Yes.
starting. That's good to know. If we move on then to the PBMC isolation part, do you have any tips that you'd give to people who are doing these processes maybe for the first time?
Yes, the PBMC's isolation requires some manual skills and it will get better with experience. But for beginners, I'll suggest to use an electronic pipetter at the slowest possible speed to stratify the blood on the cycle because it's really, really important to stratify the blood samples really slowly and with a constant flow. So maybe if you're not used to uh, do it with like a Pasteur pipette, using an electronic pipette or make it easier. And some other critical steps are like retrieving the buffy coat, that is the white thin layer between the plasma layer and the fical layer after centrifugation. And for this part, I suggest to remove some of the plasma layer to make easier to reach the buffy coat. And so it's better maybe to take some of the plasma layer. I say the plasma, but you already removed the plasma. So it's the remaining part of the plasma and some of PBS that you used to resuspend your samples. So it's better to take this part than to sink your pipette into the fical layer because the presence of fical can increase your sample contamination. Also, when you have to resuspend the samples with the 5ES, it's important to do it carefully and to make an homogeneous sample because if you don't do this well, the sample will not stratify on fiber well. And if this goes wrong, you will have again the contamination from red blood cells that you can remove with a red blood lysis buffer, but it will compromise your isolation. We will have less cells and your experiments could give you different results. So it's important. It's an easy protocol, but it's important to do it carefully and with attention because every steps can increase the risk to compromise your results of future analysis.
I really like that you summarized this with like just performing the experiment carefully and with good attention. And I think that's something that people should keep in mind probably for every protocol.
Yes.
All
So
right, Gerardo. Thank you so much for meeting with me today. This was really helpful.
Thank you.